So human fertilization, implantation, and amniotic development. Let's have a look. We know that this slide, all this is really saying, right, is that, right, human fertilization really is about combining the spermatozoon with the secondary oocyte to produce a zygote. All right. So it's initiated by the penetration of the sperm, right, through the zona, through the is it the zona pellucida, right? And then when the genetic information converges, it becomes a zygote, which is a single and diploid cell. And what's going to happen is that the zygote is going to undergo rapid division and development, and then it's going to be implanted. And after we implant, now it's going to actually go through more development, and then we stopped up when we look at the fully formed amnion. That's where we stop in Cape, all right? So we're gonna look at that entire process. So I just put these headings here because it helps me to lead you through it, right? So let's start here. So we have ejaculation occurring, releasing sperm from the male reproductive system into the female reproductive tract during sexual intercourse. Once inside the female reproductive tract, the sperm will undergo capacitation, and we know that capacitation is the biochemical process, right? That causes physiological changes that render them, right, the sperm capable of fertilizing an egg. Capacitation does that, and it also increases the amount of motility. Capacitation occurs as a sperm interacts with the female reproductive environment, including the cervical mucus and the fluids within the uterus and fallopian tubes. So during capacitation, right, sperm undergoes alterations in membrane fluidity and surface protein composition, enabling them to penetrate the egg's protective layers. Lovely. That is what capacitation is. It gives the sperm the capacity to do its job. All right. So after ejaculation has occurred and they have entered the vaginal canal, right? The vaginal muscles will start. Well, after copulation, right? There's going to be a rhythmic contraction of vaginal muscles, so there's going to be some tension there, right? So there's going to be some muscles contracting, right? And it's going to aid the movement, or it may aid the movement of the sperm towards the cervix and the fallopian tubes. So these contractions help to propel the sperm through the female tract, right? So you're gonna have some contractions there, right? So note now, we're skipping to where the female is there, right? Now ovulation. Now we know what ovulation is specifically. We spoke about ovulation, right? So after ovulation, now we know that secondary oocyte is there chilling in the fallopian tube. It's there, all good, right? Wondering if it will have a visit soon, right? While the sperm navigate through the cervix, right, with its lined with cervical mucus, right, which becomes more hospitable to sperm during ovulation. Remember, during ovulation, what we said about the cervix. The cervix produces mucus, but within ovulation, it produces a less viscous mucus, right? A less thick mucus, right? So even though the sperm will be coated in the cervical mucus, right? It will be more conducive at the time of ovulation. So cervical mucus would act as a conduit for sperm, allowing them to pass through the cervix and enter the uterus, right, en route to the fallopian tube. Okay? Sperm release. Now, let's look at it. Sperm approaches the egg in the fallopian tube. They undergo the acrosome reaction. So the acrosomal vesicle releases enzymes, right? And different types of mixtures, right? So from the tip of the sperm's head, right? It it will release these acrosomal substances and enzymes, right? To help dissolve the protective layer surrounding the egg, the zona pellucida. Hyaluronic acid is one of the substances released from the acrosome during the reaction, aiding in the breakdown of the zona pellucida and facilitating sperm penetration into the egg. Now, there's one thing to note: the egg knows what it's doing. So after the sperm actually penetrates, the zona pellucida gets much harder, right? So that it prevents polyspermy, right? So it prevents multiple sperm from entering. So after one enters, the egg closes off. The egg stops. No amount of acrosomal information would actually like cause it to break down like that anymore, right? Because what's happening, is that the sperm attaches and allows for the acrosome to be activated, releases enzymes and acid, right, and breaks into the egg. After that, everything becomes extremely hardened and it doesn't allow any other sperm to come. So first come, first serve. Combination of genetic material forming. So zygote. So once sperm penetrates the egg's protective layers, the genetic material from the sperm containing a single set of chromosomes, so it's haploid, fuses with the genetic material of the egg, which is also haploid, right? So this fusion forms a zygote, right? So it's important to know when we actually have the sperm lodging onto the egg, meiosis will continue and meiosis will be completed, and then that haploid number, right, that is in the egg will now bind with the haploid number for the sperm to create a diploid zygote, the initial stage of embryonic development, which is now a single diploid cell containing a complete set of chromosomes from both parents. So this is where we all began. The zygote undergoes rapid cell division and development has developed, right? It undergoes rapid cell division and development as it travels down the fallopian tube towards the uterus, where it implants into the uterine lining, initiating pregnancy, right? So let's look at what's happening now, right? We look at fertilization there. We spoke about the traveling throughout the entire thing, what happened throughout every single stage, and that fertilization has now occurred. We know our zygotes. Now, when we look at blastogenesis and implantation, we're going to look at what happens after fertilization. Are there any questions or concerns with anything I discussed here? Okay. So if there are any questions, feel free to ask even after I finish the PowerPoint, right? So questions are important. So blastogenesis and implantation now. Fertilization occurs. We know that fertilization has occurred, right? Spoke about it. Now, following fertilization, the zygote undergoes rapid cell divisions called cleavage, right? These divisions produce multicellular structures known as the morula, right? And consists of a blast of balls of cells, right? We can refer to it as a blastula sometimes, right? So it's a morula mostly, right? A cleavage, right? The morula, right? Which continues and the morula undergoes further cell divisions and compaction. So it's compact balls of cells that we're creating, right? Resulting in the formation of a blastocyst. The blastocyst consists of two distinct cell populations, the inner cell mass and the outer layers of cells called the trophoblast. Right? Now the trophoblast differentiates into two layers, the inner cytotrophoblast and the outer syncytotrophoblast. The syncytotrophoblast is a multi-nucleated layer formed by the fusion of the trophoblast cells. These trophoblast cells play a crucial role in the implantation by invading the uterine lining. Then we have implantation. Blastogenesis, implantation, implantation occurs when the blastocyst attaches to the endometrial lining of the uterus. The process of implantation involves several steps, right? So I think we should scratch that. I need to edit this PowerPoint. This shouldn't say blastogenesis. This should just say implantation. All right. So it should probably say after blastogenesis. It should be after blastogenesis, implantation occurs, right? So we have these four different stages for implantation that I placed into this PowerPoint. So apposition, the blastocyst comes in contact with the endometrial lining. So apposition just means it touched the lining, it touched the endometrium, right? Adhesion molecules on the surface of the trophoblast and the endometrial cells facilitate attachment between the blastocyst and the endometrium. Meaning, say, in apposition it touched the endometrium, in adhesion it combined itself with the endometrium, right? Then we have invasion. Trophoblast cells from the blastocyst invade the endometrial stroma, anchoring the blastocyst to the uterine lining, right? Now we're actually invading it, right? And then we have decidual reduction. Implantation triggers changes in endometrial cells, leading to the formation of a decidua, a specialized layer of cells or tissue, right, that provides support for the developing cells. And it is this decidua which we will look at becoming, right, or developing into our amnion or our placenta, right? So that's implantation there. So this is what is happening there. So along the way for embryonic development, we have the creation of the morula and the blastula, right? And then that will now form a blastocyst. And the blastocyst now will now be attached to the endometrium. So we have, so it comes down now, right? Remember it has the the cells in the middle, right? And then the trophoblast on the outside. It will now go through apposition where it's close to the endometrium, then adhesion where it's trying to bind the endometrium, and then invasion where it's trying to suck itself into the endometrium now. And it's surrounded by a decidua, right? That tries to help to nurture it, nourish it. Alright? Lovely. So that is how it is done. Alright? So the next thing to go on to know is the development of the placenta and the amnion, right? Which is a closer look into embryology. Any questions or concerns? For what we just did a while ago? Going once? Going
Sir,
twice?
can you
Go ahead.
explain this diagram again, please? Sir, can you
Okay,
explain this diagram again?
so with this diagram, Cape doesn't go this far, so we're not going to explain what a gastrula is really, but we're really Cape really pauses us here, right at the blastula. So remember, fertilization will occur, and after fertilization occurs, right, we're gonna have rapid development, right? So rapid mitosis, right, creating what we call a morula, and will develop into a blastula. Right, so inside the blastula you're going to have, remember what we talked about over here, right? Inside the blastula, right, itself, well, the blastula itself will become a blastocyst, right? And what the blastocyst is really is a amount of inner cells, right? And then the trophoblast on the outside, right? So you're gonna have some inner cells in the middle and you're gonna have a trophoblast on the surrounding, right? So this is what's happening over here on the bottom diagram here, right? And then what will happen now is that that blastocyst will come close to the endometrium, and that would be called apposition. When it now tries to use trophoblastic cells to bind to the endometrium, this is what we call adhesion. And then when it starts to know, sorry about that, when it starts to know, what we'd say now, attach itself or try to invade the endometrium, right? That is what's happening there, the invasion, and then we're gonna have the creation of the decidua in the middle. All right, so that's what's happening there. Okay,
Sir, your screen stopped sharing.